Okay, hi, I'm Valerie Brogdon and I manage the FIB lab at CAMCOR at the University of Oregon. I joined CAMCOR a year ago and that was around the same time we got our fancy new plasma FIB. And the exciting thing about this tool is it has the option to switch between 4 different ion species. So not only do we have xenon, we also have the ability to use nitrogen, oxygen, and argon. Here's what the UI looks like. It's similar to any other modern Helios, except for we have the ion switching tool built into the UI right here. And it takes about 20 minutes to switch between gases and get the lines purged and the gas stable and ready to use. So what can we do with these different ion species? Well, we've been trying to figure that out. We at CAMCOR are very early adopters of the Hydra, and there are only a few in the world. So we've been doing a lot of work to characterize the different ion species and figure out what they're even good for. There is a lot of evidence that oxygen is good for milling organics. And here's a side-by-side comparison of a cut that I made into a mouse retina with both xenon and oxygen. Xenon on the left, oxygen on the right. And you can see that's just a night and day difference, with oxygen being the clear winner. And here is an example of some work that one of our physics PhD candidates did in our lab. And what he's doing is milling lenses into diamond. And you can see that the oxygen cut on the right is just much smoother and more artifact-free. And I really want to go back to this mouse retina and show you this movie that I made because it came out just beautiful and I'm really proud of it. And it's a few minutes long, so I won't make you sit through the whole thing, but you can find it on my YouTube channel. I will skip to the end so you can see this 3D reconstruction. Oops, let's go back a slide. 3D reconstruction I made of the individual blood cells inside of the capillary. And this came out really nice. So this is a slice and view I did with the oxygen beam. And one thing that I want to point out about this slice and view is that it is about this slice region came from 90 microns below an extremely topographical surface, and there are still no curtaining artifacts. The slices came out just beautifully. So we've got some ideas for oxygen, but what about the other ion species? Well, there isn't a clear application yet. In order to figure out what to do with them, my lab entered into a collaborative study with Thermo Fisher to characterize the sputter rate of several materials using different ion species at different energies. And I automated this whole experiment into an iFast recipe that can be run by anyone who has a Thermo Fisher tool. All you need is the iFast runner, and that comes installed on every tool. You don't need to buy anything special. You can't edit the recipe unless you have the developer's kit, but you can run it, and we want you to run it. So the first thing the recipe does is it asks the user which substrate they're using and which ion species they will be milling with. And this is for data organization and naming purposes. Next, the recipe asks the user to navigate to the Faraday cup, set eccentric, focus the FIB, and zoom all the way in. The recipe repeats this process for each beam current, and the measured current and nominal current are written to file. The beam conditions we are using are shown here. The nominal aperture values for current vary from one ion species to another, and the script just selects the closest available aperture for the current that was established for xenon. So that's why they're all different. We wanted them to be the same, but the aperture strip is a discrete thing, and it's going to create different beam currents for each different ion species, and that's just the way it is. Yep, so those are the conditions used. The next thing the recipe does is an optional spin mill step. So it drives to the substrate to be milled, tilts the stage to 52, and mills a large circle of radius 50 microns. And then what it does is it tilts the stage to -10 degrees, and it images with a large beam current and rotates the stage, and continues that process over and over again until all of the gunk is just cleared away. There's no oxides or dirt, and we're left with this clean surface. So next up, the recipe drives the stage to a clean region within the circle, and it asks the user to input the time that's going to be spent milling. And then we start milling boxes. So there's a loop for each beam current. The stage automatically moves to the correct place after each mill, and we end up with a grid with energy increasing in the Y direction and milling time increasing in the X direction. Now the recipe mills lines on the rightmost box of each row, and this is our measurement tool. So what the user is going to do is place a cross-section corrected rectangle measurement tool on the outside edge of the line and the inside edge of the line. And that's how we know how deep the mill crater is. So these results get re-input into the recipe and written to file organized by material type and beam conditions. So what were the results? Well, it really is quite different for each of the ion species. It varies by an order of magnitude for some of them, with xenon being the clear winner for silicon volume removal. And one thing that we thought was super interesting about the sputter yield of xenon is that it steadily increases, but some of the other species like oxygen and nitrogen have a clear peak at low energies. So what's going on here? Our hypothesis was that maybe we're changing the hardness of the silicon as we mill by creating silicon nitride. So we wanted to do an experiment to check on nitrogen implantation into silicon. So we set up an experiment to test this. First, we do the spin mill step that I described earlier, where we clean off the oxides, and then we mill a 500 micron square, and then analyze that milled region with XPS. We mill that square with nitrogen, obviously. So what we found when we put this milled square into the XPS was that we got a peak here at 397.7 EV. And that is the characteristic peak for a nitride. So we are in fact forming silicon nitride as we mill with nitrogen, and silicon nitride is harder than just silicon alone. So that's the question. If we are hardening the substrate, can we utilize the formation of silicon nitride to our advantage? Like maybe we could use it as an etch mask, a protective cap. There's all kinds of interesting things you can do with selective hardening. To test the application of potential selective hardening, oxygen was implanted into silicon with a range of doses to alter the chemical reactivity. After oxygen implantation, the sample was exposed to xenon difluoride, which attacks native silicon, and regions on the left of this image were implanted with more oxygen. You can see that they completely resisted the chemical attack, whereas the lightly implanted and non-implanted regions were etched. So there could be a lot of interesting applications here for selective hardening of material with an ion beam. Next up, we have titanium. And one thing that I found very interesting about these results is that I would have expected xenon to be the fastest miller for most materials, but for titanium, gallium is the clear winner here for fast volume removal. And some of the most interesting textures resulted from milling with novel ions on titanium. We found that oxygen milling forms a unique swirling pattern like you see here, which seems to nucleate around scratches on the substrate. We thought it was so weird that we did it again, and it happened again. So it's pretty clear that some sort of chemical reaction is taking place on the surface during milling, and we don't understand what it is. So further studies will include characterizing these features chemically using TOF. So next up we have diamond, and I already talked a bit about oxygen milling on organics, but I wanted to use the results that we generated on diamond to inform our speculations about what's going on with oxygen milling on organics. So the sputter yield on diamond is higher with oxygen than most other species, except for xenon and gallium, of course, which are higher than everything always. In addition, while the other milling species show a drop-off in sputtering efficiency at the lower energies, the change in yield for oxygen is not strictly decreasing. In fact, the sputter yield for oxygen seems to level out as beam energy decreases. And we speculate that that's because the oxygen combines with the carbon in the substrate to form a volatile carbon monoxide or maybe carbon dioxide species. So there's a chemical removal mechanism in addition to just the mechanical sputtering component. And this chemical volatilization is active even at the low beam energies. So we think that's why oxygen cuts look so nice on organics and why the mill rate stays fairly high at low energies while everything else kind of peters off. And the final substrate that we tested is LR white, which is a fixative for biological samples. And I found that for most of my beam conditions, the results just looked absolutely terrible. And there isn't even really a point in measuring that because it looks like Bryce Canyon. Except for nitrogen. Nitrogen ended up making a nice, smooth, artifact-free mill on LR white, and nobody knows why, really. I don't even know if that's repeatable, but that's what we found in this experiment. The nitrogen cut looked really nice. Everything else looked terrible. So all of these results can be found in an online database. It includes all the measurements we took and all of the images for each material and each beam condition. And you can find it right here. You can click on this link. And we invite all of the FIB community to use our recipe and collaborate with us and add to the database. The vision is that this can be a shared resource for any researchers who want to know which ion species and which beam conditions they should use for specific material types. So the more information we can get in the database, the better. Thanks a lot. I'm now available for questions.